Found in gluten 13. Okay, gluten is a protein found in crops that can cause human health problems. Scientists are breeding barley cultivars to produce ultra-low gluten levels. A commercially produced barley, which is sloop, and a low gluten cultivar, LG, were crossed to produce two different cultivars with an ultra-low gluten levels, ULG1 and ULG2. Okay, the gluten content of each cultivar is shown in the table, and you have this here. Calculate how many times greater the gluten content, very posh form of gluten, content of sloop is compared to that of ULG2. And so we're just looking at this compared to that. So 57 divided by 0.004, which may give you a number which you think must be too big, but really that should not actually really surprise you. You know, this is 0.004. And you've got 57 in the top, so yeah, that's how many times greater. The allele for ultra-low gluten is recessive. To investigate if the cultivar LG was heterozygous for gluten, it was crossed with the cultivar ULG1, which was homozygous for this recessive. Okay, they name this type of cross. Okay, this is a test cross, because we have something that we know and something that we're trying to work out. Okay, describe the expected phenotypes of the offspring if LG was heterozygous. Okay, so if it was heterozygous, okay, you can do a little Punnett Square if it makes you feel better. Okay, we know that this one, the ULG1, was homozygous recessive for gluten. So let's go little g's. Okay, if it was heterozygous for the LG, then we would have a big G little g. So we'd get a big G little g, we get a big G little g, and a little g little g, and a little g little g. Okay, so we would have half of them would look dominant, so not with the ultra-low gluten, and half of them would. Okay, 50-50. That's what you need to say for that one. Okay, spin down a little bit. Right, barley is a naturally inbreeding plant. Explain why inbreeding depression would be unlikely to be a problem when a barley cultivar self-pollinates for many generations. Okay, so if you have inbreeding depression, that's because you have a build-up of deleterious alleles. But if it's a naturally inbreeding plant, they've been doing this for a while, and that means that any time when they have built up those deleterious alleles, that's then been selected out. You know, like natural selection has kicked those ones out, they've been less effective. And so the ones that haven't had the build-up, so less of the deleterious alleles, will just continue to self-pollinate. So basically, over time, they have bred out the deleterious alleles that would normally be a problem in inbreeding depression. Okay, it's quite a nice one to talk around. Okay, D. Barley grains contain the enzyme amylase, which breaks down starch in the grain to sugar used in brewing beer. Okay. Average grain mass, starch content, and amylase activity for 3 barley cultivars are shown in the table. So we've got our 3 different cultivars, the average mass of a single grain in milligrams, and then how much starch in each grain as a percentage, and the amylase activity in units per mg. Okay. As well as total mass of all the grains, state the information required in order to calculate the average mass of a single grain. Well, I would need to know the number of grains. It's an odd question. Just checking if you can read it. Select a cultivar from the table that would be best to use in beer production and justify your selection. Okay, so it says barley grains contain the enzyme amylase, which breaks down starch in the grain to sugar used in brewing beer. So I'm going to break down starch to sugar. Okay, so I can totally see you picking sloop and giving because it has the highest percentage of starch content in the grains. Or you could have gone with ULG2 because it has the highest amylase activity. So although there's less starch, it would break it down faster. Either of those is fine, but you have to back up your justification. And